This is a revision video on the Meselson and Stahl experiments. So there are 5 revision questions. Pause the video and complete those questions, and then once you've done that you can continue the video and check your answers. Okay, so let's take a look at the answers. So your answer for an isotope should be something like this. And so in the case of Meselson and Stahl, if we consider a DNA nucleotide, so this is a very basic structure of a DNA nucleotide that you should be able to draw. What Meselson and Stahl did was to use isotopes of nitrogen. So if we look at the structure of that base in more detail, so this is adenine as an example, we can see those nitrogens in the adenine, and in this case they're nitrogen 14. So nitrogen 14. So what Meselson and Stahl did was to give bacteria either nitrogen 14 that they could use to build their bases in the DNA nucleotides, or they gave bacteria the isotope of nitrogen 14, which is nitrogen 15. So they ended up with 2 cultures of bacteria, one that had nitrogen 14 in its DNA and the other that had nitrogen 15 in its DNA. And they could consider the DNA that was made in these 2 circumstances either light, so DNA that contained nitrogen 14 was considered light, and the DNA that contained nitrogen 15 was considered heavy, because all the nitrogen had an extra neutron. And so when the double-stranded DNA was made, you would have light double-stranded DNA from the light bacterial culture and heavy DNA from the heavy bacterial culture. And so going forward in this explanation, we will use these diagrams as double-stranded DNA. Purple is light and red is heavy. So let's move on to question 2. So a centrifugation is this. And so if we just give an example diagram, so this is a very, very basic diagram of a centrifuge. There are 2 tubes. It's a machine, and it spins these tubes at high speed. And when it's spinning, the heavier substances in the solutions in those tubes move further down. So we can separate substances by size. And so this was used in Meselson and Stahl's experiments to separate out light and heavy DNA. So you can see in the diagram here, centrifugation of the DNA from bacteria grown with light nitrogen and heavy nitrogen, so they create light DNA or heavy DNA, it would separate out like this. And so that's a useful way of seeing or measuring the type of DNA that's made. So let's go on to question 3. So they were trying to find evidence for whether DNA replication was conservative or semi-conservative. Question 4. What did they find? Well, the first thing they did was to take some bacteria and culture it with heavy nitrogen. So all the bacteria in that culture had heavy DNA. And if that was centrifuged, you would get this result in the centrifuge tube. Then what they did was to give that culture of bacteria light nitrogen. So any new DNA that was made by that bacteria would use DNA nucleotides that contained nitrogen-14, light nitrogen. So when that DNA replicated, we call that generation 1, so one round of replication, we can look at two possibilities that could have occurred. If DNA replicated in a conservative manner, then the new strands would contain light DNA, and if it's conservative, both new strands would join to make a new double-stranded DNA, and the original heavy strands would stay together to form the other strand of DNA. And if we looked at that in the centrifuge tube, after centrifugation, we would see this. We'd get a light band and a heavy band. If, on the other hand, it was semi-conservative, Meselson and Stahl would have observed this result. So an original strand combines with a new strand in both cases, and so you get a heavy strand and a light strand combined together after replication. And the result in the centrifuge tube would be this. You would get a mixed band because you've got both a heavy and a light strand in the DNA. And it's this that Meselson and Stahl observed. They observed semi-conservative replication. So finally, let's move on to question 5. So if Meselson and Stahl continued the replication, what would they observe at generation 3? So we know what we get with generation 1. We get a heavy and light chain. And if we allow that to replicate with light nucleotides, this is what we'd get. So that heavy strand would produce a new light strand, and that would combine. The light strand would produce a new light strand, that would combine, and the same with the other strand. And so in the centrifuge tube, we'd get this result. We'd have a mixed strand, but we'd also see the light strands. And so then if we carried on with the replication again, in generation 3, we would observe this. And so any new strands that were made would be made with the light DNA nucleotides, and so we'd get this pattern. If we looked in the centrifuge tube, we would see a similar result, but we'd obviously be getting more of the light DNA. And so in terms of ratio, you can just count it there, and what you should get is a light to mixed ratio of 3:1. So that is the revision questions for Meselson and Stahl experiment. Remember to read the textbook and revision guide on this to strengthen your knowledge, and then test yourself using those questions in those books. I will provide the pages that you need to go to in those books in the description below.